Hello, everyone. Thank you for joining and welcome to today's webinar, Cancer Immunotherapy and the Application of In Vitro Immunoassays in Drug Discovery. We look forward to an engaging discussion. This is Wendy Liu, Associate Director at Uchia Tech from Research Service Division, and I will be moderating today's webinar. Now, I would like to share a few words about Uchia Tech, our host for today's webinar. Uchia Tech provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. As an innovation-driven and custom-focused company, Uchia AppTec helps our partners improve the productivity of advancing healthcare products through cost-effective and efficient solutions. We are pleased to welcome Christine Lowe from Nimbus Therapeutics and Diego Mariscati from Phillip International. Dr. Christine Lowe is the Senior Vice President and Head of Biology at Nimbus. Prior to joining Nimbus, she served as Vice President of Translational Medicine at Chimera Therapeutics. Christine also has over 20 years of experience as a leading researcher at Citrus, Pfizer, and ICOS Corporation, advancing several programs from concept to IND in autoimmunity, oncology, and metabolic disease. Dr. Diego Mariscati is the high-content screening manager in the System Toxicology Department of Phillip International Research and Development. After completing his PhD in oncology and molecular pharmacology at the University of Ferrara, Italy, he continued his research in the tumor immunology field. He joined Phillip International in 2012, bringing his knowledge for cell biology and imaging to contribute to set up the cellular research lab. Since then, he is leading a team of researchers who perform compound profiling with high-throughput systems. Our presenter today will be Tina Wang from Wuxi AppTec. Dr. Wang trained as an immunologist and developmental biologist at Hong Kong University of Science and Technology. Her study focused on the development of microglia as well as immune response of peripheral macrophage. Having joined Uchia Tech in 2017, Tina now holds the leading responsibility for the in vitro immunology platform, providing candidate screening and mechanism of action studies, and also in IND-enabling projects to global biotech and pharmaceutical companies. I will now turn things over to Tina.
Thanks, Wendy, for the introduction. Morning, everyone, and thank you for spending time with me for the talk today about cancer immunotherapy and the application of in vitro immunoassays in drug development. I would like to start with the most updated report for global cancer progress from AACR. As shown in the figure on the left, the devastating impact of cancer is predicted to grow significantly in the coming decades, in part due to overall population growth and the expansion in the segment of worst population aged 65 and older. The increasing global cancer burden is spurring the development of cancer care as well as related scientific research. Currently, the 5 pillars for cancer therapy is well acknowledged, including surgery, radiotherapy, chemotherapy, targeted therapy, and also immunotherapy. The development of cancer immunotherapy can be traced back to the year of 1891, when Dr. William Coley treated tumors by injecting cancer patients with bacteria and reported tumor regression in many cases. However, the immunotherapy was not applied until the next century, when the benefit of interferon alpha and interleukin-2 was discovered. And these 2 cytokines were approved for cancer therapy in the late 1980s to 90s. It is worth noting that the cancer vaccine concept also came into application in the year of 1999, when Canada approved the very first cancer vaccine, Malacin, for the treatment of melanoma. Then 10 years later, the first cellular immunotherapy arose when the FDA approved Poland for the treatment of prostate cancer. Despite this FDA approvals, immunotherapy was not widely used for another 10 years until the immune checkpoint antibodies, anti-CTL4, anti-PD-1, and anti-PD-L1 came on the stage, and the impact of immunotherapy was highlighted by Dr. James Allison and Tatsuko Honjo being awarded the 2018 Nobel Prize in Physiology and Medicine, which inspired the further development of immunotherapy for cancer. At the end of 2018, as many as 7 immune checkpoint inhibitors were approved by FDA for the treatment of cancers, and all of them were immune checkpoint blockers against the PD-1, PD-L1, or CTL4. The investigations for immune checkpoint inhibitors have moved far beyond that, and today we're really looking forward to the next generation of immune checkpoint inhibitors. In addition, cellular immunotherapy also achieved another milestone with approval of CD19 CAR-T for B-cell lymphoma in the year of 2017, which took us into the era of cell therapy for cancer. With the rapid development of immunotherapy in the past decade, currently the approaches for cancer immunotherapy have expanded into 6 categories. First one is target antibody-driven therapy. The classical mechanism for antibody therapy is by binding on the antigen on cancer cell and remove cancer cell by ADCC, CDC, or ADCP. In addition, antibody drug conjugate, which is one of the fastest growing anti-cancer drugs, built by conjugate anti-cancer drug with antibody to achieve cytolytic cancer specifically, and also bispecific antibody engaging 2 targets in 1 molecule, highlighted by the approval of CD19 CD3 BiTE in 2014 for B-cell precursor ALL. The second category is immune checkpoint. It is well known that a vast pool of immune checkpoint targets beyond PD-1, CTL4 are under development. Both literature and the clinical outcome has demonstrated that treatment of PD-1 antibody will lead to the increased expression of other immune checkpoints like TIM3 and LAG3, so that combination of different immune checkpoint inhibitors became a trend for current clinical trials. The third one is cancer vaccine, which is to training the immune system to recognize and attack cancer. There are both preventing cancer vaccine like HPV for cervical cancer and also therapeutic cancer vaccine like Macrophage for prostate cancer. Next one is adoptive T-cell therapy, which is highlighted by the approval of CD19 CAR-T. But besides CAR, the next generation cell therapy, including TCR-T and the TIL therapy, arrives as hotspots with different advantages. Since CAR-T can target only surface single antigen expressed on tumor, while TCR-T can respond to intracellular antigen tumor. But both the CAR and the TCR-T are currently limited in their function for solid tumors. And the TIL therapy as a complement for that, which can recognize multiple tumor targets and have potential to kill solid tumors. Then oncolytic virus, which serves as a driver to lead to tumor cell lysis and release tumor antigen to amplify the immune response and facilitate immune cell infiltration. So combination of oncolytic virus with cell therapy have potential to kill solid tumor. The last one is immunostimulus. Mainly refers to cytokine like interferon gamma, interleukin-2, which is to stimulate immune response broadly. So for benefits and the disadvantage of different immunotherapy approaches, I would like to invite Christine to share with us for her comments.
Thanks very much, Tina. I think your summary nicely illustrates the expanding variety of immunotherapy approaches and also the fact that there's room for improvement in these IO therapies. So each of these approaches has provided efficacy, but none on its own is a magic bullet. So further, not every tumor responds equally well, as we know. And not only do the mechanisms of action differ for each of these approaches, but there are also different considerations for manufacturing. So a lot more exciting things to happen in the field. I'll turn it back to you, Tina.
Thanks, Christine, for the insightful comments. With better understanding of the immunotherapy approach, in the next slides, I would like to talk about immune surveillance during tumorigenesis for better understanding the mechanism of cancer immunotherapy. The battle between immune system and the cancer cell could be broadly divided into three phases: elimination, equilibrium, and escape. Each cell is estimated to experience over 20,000 DNA-damaging events each day, which will normally be repaired by DNA repairing mechanisms. Cells with the DNA that is not properly repaired will become abnormal and will be recognized and removed by immune system. This is elimination phase. If an abnormal cell survives the initial elimination attempt but is not able to progress to cancer, it's being maintained in a state of equilibrium. With the selection pressure of the immune system and the accumulation of mutations, finally, the cancer cell equipped with the ability to avoid the recognition by immune cells and escape the immune surveillance, and it becomes clinically detectable cancer. The mechanism that the cancer cells take to avoid the immune system include reduced antigen presentation function, reduced T-cell activation, recruitment of suppressive immune cells, and also increased expression of inhibitory immune checkpoints. To further zoom into the tumor microenvironment, there's much crosstalk between immune cell and the tumor. So starting with the tumor cell releasing antigen, and the antigen being processed by dendritic cells, then the antigen-loaded DC will further present the tumor antigen to CD4 and CD8 T cells. The activated CD8 T cell will recognize cancer cell and perform cytotoxicity, and activated CD4 T cell will release cytokines to further enhance the function of CD8 T cells. On the other hand, the CD4 T cell could also activate the B cell to eventually become plasma cells that secrete antibodies that target the tumor antigen. Once the antibodies bind into the tumor cell could be recognized by NK cell or macrophages and removed by ADCC or ADCP process. On the other hand, tumor cell will release chemokine cytokines to recruit immune suppressor cells like regular T cell or MDSC, which will inhibit immune function and promote the tumor progression. In the following slides, I would like to dissect each of the above process and combine with introduction of WuXi in vitro immunology capability to demonstrate how we can provide help for IO drug discovery. So starting from the first event, when a DC processes the tumor antigen and activated T cell. Well, T cell require two signals to become fully activated. One is provided by T cell interaction with MHC presented antigen. The other is co-stimulation signal mediated by CD28, CD86, CD8 expressed by dendritic cell. And beyond the two classical signals, there are a variety of immune regulators involved in the crosstalk between ADC and T cell. So here we have established the MLR system, which is in vitro allogenic DC active T cell model. We take advantage of this system to study the function of tested compounds on the crosstalk between DC and the T cell. Since PD-1 is highly expressed on T cell and PD-L1 is expressed on dendritic cell, so in this model, we use PD-1 antibody as a control, and we demonstrate that after blocking PD-1 signaling, T cell activation could be significantly promoted. And here we look at the proliferation and also cytokine secretion by ELISA. So from the figure, you can tell that as we increase the PD-1 concentration and further increase the proliferation and increase interferon gamma and interleukin 2 in the supernatant in a dose-dependent manner. So the lower panel here is to show that we have experience to screen the combinatory effect of PD-1 antibody with other small molecules and we find our target compounds which display a significant synergistic effect with the PD-1 antibody. In addition to DC-activated T-cell model, we also use antibody to achieve T-cell activation. So this cartoon here nicely illustrates the molecular mechanism involving in T-cell activation. So you can see that CD3 is the downstream of TCR. So here we use CD3 coupled with the CD28 antibody to induce robust T-cell activation. And we analyze on first is T-cell proliferation, then the cytokine secretion by ELISA, and also CD25 expression on T-cell surface. And here is to show that one of the compound we screen out, which is display promoting effect on T-cell activation for different readouts. Besides antibody and cell-mediated T-cell activation, we also have established superantigen-based T-cell activation model. And here, the superantigen we use is SEB, which is toxin produced by gram-positive bacteria. The mechanism underlying is that SEB could simultaneously binding to the class II major histocompatibility complex and the TCR at the variable region of the base chain. As this binding event occurs on a subset of T-cell Wee-bit domain, so around 20% of T-cells could be activated by SEB. And in addition, the immune checkpoint inhibitors also involved in the crosstalk between T-cell and the dendritic cell. So here, again, we use PD-1 antibody as control in this assay. So from the curve, you can tell that as we increase the concentration of PD-1 antibody, it increases interferon gamma and also interleukin-2 in a dose-dependent fashion. Within tumor microenvironment, CD4 T-cell is highly versatile and it can polarize into different subtypes. Currently, the most well-studied T helper subtypes, including inflammation-promoting Th1 cell and anti-inflammatory Th2 cell, immunosuppressor regular T cell, and also Th17, which is involved in autoimmune and inflammatory disease. We have established in vitro T helper differentiation assay, which outside from naive CD4 T cell enriched from PBMC, and then differentiate using different cytokine cocktail combined with the CD3, CD28 stimulation. And compare with Th0 cells, from the figure you can tell that successfully differentiated Th1 cell exhibit highly expressive interferon gamma, and the Th2 cell exhibit high expression of IL-4, and Th17 cell increased expression of interleukin-17A, and the regular T cell increased the CD25 and FOXP3 double positive population. CD8 T cell was the major effector cell within the tumor microenvironment. To study the function of CD8 T cell, we have established assay in both mouse and human. This slide shows the OT1 CD8 T cell based killing assay. The OT1 mouse has been engineered to express TCR that recognize a specific OVA peptide presented in MHC. And the OT1 system was widely used to study antigen-specific T-cell response. In this assay, we first collect OT1 mouse melanocytes and stimulate with OVA peptide for 3 days to enable the expansion of OVA-specific CD8 T-cells. After 2 days of expansion, more than 90% of remaining lab cells are all CD8 T cells. Then we pause the target EL4 cell line with OVA peptide for hours, then perform co-culture with the CD8 T cells. The readout is target cell lysis indicated by PSA. So from the curve, you can tell that OT1 T cell can elicit robust killing towards OVA-carrying EL4 cell, which is in an effective target ratio dependent fashion. For humans, we have established the CD3 BITE-mediated primary T cell killing towards target cells. And BITE is short for bispecific T cell engager, which One domain binds with CD3 to activate T cell, and the other domain binds with specific antigen expressed on tumor cell. With the help of BiTE, T cell could specifically target the tumor cell and mediate cell cytotoxicity. And here we use CD19, CD3 BiTE as control, and the co-culture of primary T cell with the Raji cell, which is highly expressed CD19 on the surface. And then we analyze T cell activation marker, including CD25 expression, T cell proliferation, interferon gamma secretion, and also target cell lysis. One of the escape mechanisms by cancer cells is to recruit suppressive immune cells like regular T cells to inhibit the normal function of other immune cells by secreting cytokines like TGF beta or interleukin-10. For studying the function of the regular T cell, we have established in vitro T regular suppressive assays as indicated in the next slide. So here we use natural Treg, which is sorted from PBMC and expanded using CD328 in vitro. Then we isolate autologous T effector cells. Then we perform co-culture of regular T cell with effector T cell in the presence of CD328 stimulus for 4 days. And we analyze proliferation of T cell and also interferon gamma by ELISA. And from the data, you can tell that as we increase the ratio of regular T cell in the co-culture system, we further decrease the effector T cell proliferation and the decrease in interferon gamma secretion as well. Well, exhaustion phenotype of T cell is frequently observed in many cancer types, where we reduce the cytokine secretion, decrease the proliferation, and also cytolytic activity. So restoring T cell exhaustion provides promising opportunities for cancer therapy. And here we have established the in vitro T cell exhaustion model to enable compound screening that reverse or restoring T cell exhaustion. So in this assay, we isolated primary T cells and continuously stimulated with CD3, CD28 for 10 days, and confirmed the exhaustion phenotype by examining the immune checkpoint marker, and also confirmed that the cytokine interleukin-2 and interferon gamma was further decreased. And we further test the function of the exhausted T cell in the MRI system. So compared with the fresh T, you can tell that exhausted T cell indeed reduce their response for DC activation. And in this system, PD-1 antibodies can partially reverse exhaustion phenotype. Speaking of targeting on T cell for cancer therapy, as Nimbus recently announced a very potent HPK1 inhibitor displaying good efficacy in vivo and having potential to be a good IO agent. I believe Christine might have thoughts on this asset for looking at compound targeting T cell for cancer therapy. Christine, would you care to comment?
Yes, thanks very much, Tina. I think all the T cells assays that you've described here are really useful for looking at an agent's effects on T cells. I would specifically highlight that the assays that test specifically for T cell function in the context of those other cells I find are especially helpful. Those assays include MLR, where dendritic cells are present, or the T cell and tumor cell assays, where one can look at the CD8 positive general killing of tumor cells, as well as killing in an antigen-specific way. So given that the tumor microenvironment can suppress the immune response, I find that the T cell exhaustion and the Treg assays that you just described are really valuable for evaluating how well an agent might act on the T effector cells to circumvent those types of immune suppression. Thanks very much, Tina. I'll turn it back to you.
Thank you, Christine, for the comprehensive comment. Currently, T cell is indeed the most popular target for drug development, but other immune cells like B cell also play an important role in mediating tumor immune response. In terms of B cell activation, there are two approaches. One is T cell independent activation mediated by extensive cross-link of BCR. The other is T-cell dependent activation, which is dependent on B-cell present tumor antigen to T-cell with the help of CD40, CD40L interaction, and cytokine release from T-cell to achieve B-cell activation. So currently, we have established in vitro human B-cell activation assay to mimic the physiological events. And here, we use anti-IgM antibody to cross-link the BCR to induce the T-cell independent B-cell activation. And also, we use anti-CD40 antibody combo with interleukin-21 to mimic T helper cell dependent B-cell activation. So the readout is B-cell proliferation, interleukin-6 secretion, and also IgG production. And here is to show one of the control compounds that display a promoting effect for B-cell activation for multiple readouts. Macrophages are highly versatile and plastic population and could polarize themselves into different subtypes and different cytokine stimulation. We can broadly divide macrophage into two types. M1, which is classical pro-inflammatory macrophage, and M2, which is anti-inflammatory macrophage. On our platform, we have established the M1 and M2 polarization assay. We're starting from CD14 positive monocytes and use interferon gamma to induce M1 macrophage and use interleukin-4 plus interleukin-13 to induce M2 macrophage for 7 days. And then we analyze surface marker expression by flow and also cytokine secretion by ELISA. As the result indicated here, that M1 macrophage secrete high level of interleukin-6 TNF-alpha but a low level of interleukin-10. On the contrary, M2 macrophages secrete a high level of interleukin-10 and also express high level of CD163 on the surface. Besides polarization assays, we also established functional assays for macrophages. Here is ADCP assay to evaluate the function of the antibody-induced macrophage phagocytosis in vitro. We induce macrophage from CD14 positive monocyte and it perform co-culture with target cell. And here we use CD20 antibody as a control. So the target cell we choose is B-cell lymphoma Raji cell line. And we have tried two different ET ratio with different concentration of antibodies. From the figure, you can see that anti-CD20 antibody could induce macrophage phagocytosis in a dose-dependent manner. Well, ADCP is mediated by Fc receptor. Beyond that, tumor cells also express don't-immune signals like CD47, CD24 to inhibit macrophage phagocytosis. And here, we use antibody to block the don't-immune signal, and showing in this figure that block both CD24 and CD47 can increase the macrophage phagocytosis significantly. In addition to regular T cell, MDSC also play an important role in inhibiting immune function. We have established the in vitro MDSC suppressive assay in which we induce MDSC from CD14 monocyte in the presence of GM-CSF and interleukin-6, then co-culture MDSC with autologous T cells in the presence of CD3, CD28 dynabeads for 4 days. We analyze on T cell proliferation and also interferon gamma by ELISA. So from the figure, you can see that there's as we increase the MDSC ratio, actually we decrease the T cell proliferation and also decrease interferon gamma release. Well, natural killer cells serve as one of the major effector cells towards cancer, and more treatment options harnessing tumor microenvironment targeting on NK. To evaluate NK cell function, we have established primary NK cell-based activation and the cytotoxicity assay. And here, the NK was enriched from human PBMC and then activated using interleukin-2. then perform co-culture with target cell. And here we choose K562 cell line as target cell. And we analyze on 2 dimensions. One is target cell lysis indicated by PS staining. The other is NK cell activation by CD369, CD107A expression on surface. So from the bar graph, it shows that we have control compounds that is increased NK cell activation and also increased target cell lysis. In addition, ADCC was an important mechanism for antibody therapy. We provide primary NK cell-based ADCC assay to study the function of antibodies. And here, again, we use CD20 antibody as control and Raji as target. We have done different ET ratio and tried different concentrations of CD20 antibodies. And the data shown here that CD20 antibody can induce robust ADCC in a dose-dependent manner. Well, inflammation is always associated with development and malignant progression of most cancers by tumor-promoting cytokine and also modulation of tumor microenvironment. We have developed both Toll-like receptor and non-like receptor related inflammation assay. And here is the demonstration for a NLRP3 inflammation activation assay based on THP1 cell line. NLRP3 activation requires 2 signals. One is priming by pathogen-associated molecular patterns like LPS to prepare the cell to express sufficient inflammatory components like pro-interleukin-1 beta, pro-interleukin-18. Then the second signal triggered by either ATP or potassium outflux to induce inflammatory assembling and activate caspase-1 and then processing pro-1 beta, pro-interleukin-18 into mature form and secrete out. And here, we use THP1 derived macrophage, and the priming use LPS followed with nigericin stimulation. Then we analyze interleukin-1 beta by ELISA and caspase activity as well. And here we use MCC 950 as control to inhibit the inflammatory activation. So from the result, we can tell that MCC 950 can inhibit 1 beta and caspase-1 with high potency. The discovery of STING in 2008 quickly expanded immunology research in cancer therapy. STING target agents such as ADU-100 has been approved for clinical trial. We also have systems to study STING agonists using either THP1 or PBMC system. And we analyze interferon beta by ELISA. So beyond human beings, we also have platforms to study the immune function in non-human primates. The left panel is the well-established immune profiling panel for cynomolgus PBMC. As shown here, with the optimized panel, we can detect T cell, B cell, NK cell, monocyte, and also T cell subsets in the cynomolgus blood. And the red panel is in vitro activation of cynomolgus T cell by SED, and the readout is very similar to humans. Last but not least, I would like to generally introduce the WuXi Oncology and Immunology unit. The whole department have around 550 scientists, and we have operating in Shanghai, Suzhou, Jiangsu, and Chengdu. So beyond the in vitro capabilities, we also have well-equipped animal facilities, including P2 animal labs. In addition, we have CAP-certified clinical pathology and FACSA labs. And currently, the partners we are collaborating with is more than 300. That is today's presentation, and thank you for listening.
Thank you so much, Tina, Christine, and Diego for an informative presentation. I see that, Diego, you already have a few questions for Tina. Would you like to go ahead and ask those?
Yes, thank you for the opportunity. And thank you, Tina, for the presentation and for providing us an overview of the WuXi in vitro immunoassays used in the context of drug development. I have some questions and I would like to start asking you to please comment on assay throughput. What is the screening compound capacity? How many and how quickly can the compound be assessed?
Thank you, Diego, for the questions. Actually, we perform assays in 96 well plate, and we can handle at least 10 plates per round. So if you do IC50 curve with 8 to 10 doses for each compound, then there will be around 25 to 30 compounds per round could be screened.
Seeing that there are multiple solutions to address one question, I was wondering if you could share with us some examples of the rationale that makes you choosing one assay from another.
Thank you for the very good question. In most cases, actually, we already know the expression pattern of the target so that we can choose the specific cell type with a high expressional target and develop the assay according to the working mechanism, either interrogating the cyclin activation or differentiation or other functions. But in the particular cases, for example, T-cell assays, we have multiple approaches to induce T-cell activation. Then how to choose among them will be a question. Then it is related to the MOA of the tested articles. For example, if the tested article is directly working on downstream of T-cell activation, then I would suggest to use antibody-mediated T-cell assay, since it is simple and a direct focus on T-cell activation. But if the target is on synapse between APC and the T-cell, I would suggest to use either SBB activation or MLR assay. And the choice will highly depend on the expression level of target on PBMC or monocyte-derived DC. Thank you.
Acknowledging the complexity of tumors in vivo, how well the cell-based systems are resembling the in vivo response? After our initial screening, how are they further validated?
Thank you, Diego. This is a question I have been frequently asked. And I have to say the answer is really case by case. Because in vitro assays simplifying the tumor microenvironment by isolation the target immune cells and using stimulus to mimic the response in vivo, which enable dissection of a specific biological process. For those cases with very significant assay window and the drug treatment, and the assay window is very consistent among different donors, then obviously we'll give more confidence that AD can exhibit nice effect in vivo as well. But since there's a huge difference between vitro system and the actual tumor microenvironment, so there's still possibility that other immune cells within the tumor microenvironment will pose either positive or negative effects for drug efficacy, and that needs to be further validated using in vivo tumor models.
And very last question. With a view to personalized medicine, can we envisage to have some of these assays being patient-tailored?
Thank you very much. This is, again, a very good point. So far, all above assays based on healthy human PBMC. So as we all know that in cancer patients, especially patients who have received certain therapies previously, the immune cell profiling would be dramatically changed compared to healthy people. So it would be provided more relevant evidence if we can use patient immune cells or patient cancer cells to study function of the tested drugs. There are two reasons that we have not established assays in the patient health section. First one is considering the working flow of drug development. The in vitro immunoassays sit at a very early stage, whereas for target validation and proof of concept. So the focus at this stage is on a generally functional study and required in a high throughput way. The PBMC from healthy donor is ready to use and source is sufficient, but the patient sample is limited, and there is huge case-by-case variation, which will not help on drawing conclusion at early stage. The second reason is that due to the regulation of government, it is hard for us collecting patient samples in clinical studies. So that is another reason that we have not established in vitro assay based on patient samples so far. But thank you, this is a very good point.
Thank you to you for all the answers. Thank you very much.
Thank you everyone for dialing in. And then thank you Tina, Christine, and Diego for a very informative presentation again. And now we are open for questions. Please submit your questions via the Q&A tab in your screen. And then we already gathered some questions from the audience, and then we would like to send those to the presenters today and have them answer those. So I guess the first question is for both Tina and Christine. So how could you connect and interpret in vitro data from mice and humans, since there is difference of immune system in those two species? Tina, would you like to comment first?
Yes, thank you, Wendy, and thank you for the questions. Without knowing the background of the project, I would like to have a general comment for the questions. In terms of the two of the species, I have to say both of them have their unique advantage. For mouse, the advantage is that the plenty of in vivo model resource, and you can do a genetic modification that generates a specific mouse strain. But for human, the advantage that for sure is a species advantage. But the disadvantage is that so far the preclinical human assays based on PBMC, but we know that peripheral blood, the immune environment is much more simple than the tumor microenvironment. So have potential that cannot fully demonstrate the working mechanism. So, and in other cases that for human and mouse in vitro assays, different assays have different mechanisms. So it really depends on the project itself. Yeah, thank you.
And Tina, maybe I'll add to that. Yeah. I agree with everything that you said without knowing anything about the specifics behind the question. I would say that I would even back up a step further. I don't know what the target of interest is here, for example, but there have been species differences. How well conserved is the mouse and the human target of interest, and has the test agent, compound, whatever that is, been tested against the human and the mouse target, if that's possible, in an in vitro or recombinant setting, first to make sure that that agent is behaving the same way on the target in the two different species before even taking it to the cell. So I've had a couple of experiences where the targets were not necessarily as well conserved, and so there was actually a species difference even at the recombinant protein level, then of course then translated into the cellular environment, because then the mouse cell potency, for example, in an assay would be different from that in a human cell assay potency. So I would start from there. But you know, the person who's asking the question is correct that there are some subtle differences between mouse and human immune cells. Generally, I would say that the assays that you've described here tend to be mechanistically based. So there should be some level of consistency, but again, not knowing the full details, there could be individual cases where it disconnects. Thanks for the question.
Thank you, Christine and Tina. So next question, I think for the macrophages, the pages talking about macrophage assays, what macrophages were used for M1, M2 assay? I remember that IL-4 could not stimulate IL-10 production from human BMDM and THP1 in some papers. Tina, would you like to take that?
Yes, thank you for the questions. Actually, the macrophage we use is CD14 positive monocyte we enriched from human PBMC. And for M2 macrophage, we do not stimulate mono, only use L4. We pre-stimulate with pre-culture the monocyte use the MCSF followed L4 and L13, and then turned out to be the L10 can be promote quite significantly, but not very high as the other cytokines. Yeah, thank you for the questions.
Okay, next one. So for on page 19 and several pages after that, are these bone marrow derived macrophages or human monocyte derived macrophages?
Yeah, just very similar questions. All the macrophage actually are monocyte derived macrophages, which isolated from monocyte isolated from human PBMC, and actually we have done the bone marrow macrophage, but only in mouse, but not in humans so far.
Thank you. Okay, one question for Christine from the audience. He or she said they wanted to know the secret behind stimulating proliferation by anti-IgM and interleukin-4, and that means suppression, their combination for the purpose of stimulating a proliferation should be considered for cancer immunotherapy or vice versa, because the rapid proliferation should cause rapid growth of tumor. So would you like to comment on that?
Okay, if I understand it correctly, there might be two parts to the question. One is that the individual is asking something specifically about the way the assay is set up that Tina had mentioned. So for the B cells, for instance, you stimulate using a combination of anti-IgM and IL-4 to get them to proliferate, and then I presume that the client or the person is interested in testing whether or not the test article changes that proliferative capacity, either decrease or increase. So for that one, it is purely an in vitro system where, just like for the T cell assays that you described, Tina, where you're using anti-CD3 and CD28, it's a little bit of artificial setup in that instance. So in the case of B cells, you can use IgM and IL-4 to do the similar type of stimulation as for T cells. I think there's a second part of that question where it sounded like the individual wanted to know if B cells were an appropriate target for, let's say, IO therapies. That on a very general level is certainly of interest in the field. I mean, people have certainly been chasing after T cells with immune checkpoints, for example. The myeloid population is now increasing in interest, macrophages. For B cells, there's definitely, I think, a rationale for understanding enhancement of B cell function in the tumor microenvironment, because B cells can act as antigen-presenting cells, of course, and cooperate with T cells to activate them. They can certainly also put out antibodies, which might be needed for ongoing durable anti-tumor responses. So if that's the case, then again, these sort of in vitro B cell assays might be a first step to informing on whether or not there is a B cell effect of, let's say, a test agent or article. In terms of whether or not the field is going to be able to understand if these types of agents might be good in the whole IO treatment setting, that will certainly come. So thanks very much, Wendy, and thanks to the individual for asking that interesting question.
Thank you, Christine. The next question for Tina. For your Treg cell assay, is it ready for compound screening?
Yes, it is ready for compound screening, and actually we have used the assay for plenty of compound study so far. Since all the assays were run in anti-well format, and one of the concerns may be that the regular T cell is really rare from PBMC, that you only get like a 0.1%. But since we perform the in vitro expansion, so that we can get like a 100-fold of increase in terms of Treg number, so that the number is sufficient for high throughput compound screening. Thank you.
Thanks, Tina. A couple of more questions from the audience. This one is discovering to the target cancer cells while in their dormant state, or waking them up with the classical method. Which one would you prefer?
Oh, that's an interesting question. I think that's, maybe I'll take that one, Tina and Wendy. I think that, again, is something that the field is trying to understand a little bit more of, is exactly, even with the current, let's say, immune checkpoint inhibitors that are out there that are being very closely studied in the clinical environment. It's not entirely clear yet whether the mechanism is just so-called waking up those cells that are suppressed or exhausted or energized in the tumor microenvironment, or whether or not it's the peripheral T cells or immune cells that you're then waking up in the periphery to try to get them, try to get the immune cells to then go to the tumor and attack the tumor cells. So it's an interesting question, but I don't think that there is a specific answer as yet. I think people are trying all different manners of of trying to activate the immune system. So I don't know if there's a preference in the field per se as yet.
Thanks. Next one. We know the key drivers for M2 to M1 conversion for macrophages.
Well, actually, this is mechanism-related questions. For M1 and M2 conversion in terms of the mechanism, actually, it is a complicated signaling pathway related, like related to STAT and JAK-STAT pathway, and also upstream is like toll-like receptor and NF-kappa B downstream. But there's all well studied. But generally, what we are focusing on is the different cytokine environment and related to the receptor, cytokine receptor, and related to downstream signals. And the final readout or outcome is also still the macrophage cytokine profile and also functional of the macrophage, either by promoting T cell or immune cell or inhibiting the immune cell functions. Any other compliment from Christine or from others?
No, I think I say addressed that one quite well there.
Cool.
Yeah.
So moving on to the next one. For T cell agonists, is it always showed bell-shaped curves, which make it hard to transfer in vitro results to in vivo? So what's your experience to find a proper in vivo doses? Thank you.
I might have some draw some comments, and then Christine, if you have additional comments, please feel free to add. And actually, from our experience to do the T cell activation analysis, it is not a very common phenomenon that you get a bell-shaped curve. And when we get a bell-shaped curve, first thing we will think of is consider the sensitivity of the assay. For example, if you have a stimulus within the assay, then we will think of whether the stimulus is so potent, so strong, so that, or the stimulus is so weak, so that lead to the dose response of the compound is weird. And after you optimize assay stimulus conditions, and then you have the chance to get a dose-dependent nice curve, and then follow up with the dose choice. But I have to say, when you got a bell-shaped curve, this is not something normal, and this is for sure it's hard for us to choose the appropriate concentration of the compound. Yeah.
Yeah.
Thank you for the question.
Yeah, Tina, maybe just to add on top of that, I mean, technically speaking, bring it back purely to the T cell, without thinking about the test agent or whatever it is. I mean, it is a biologically relevant phenomenon that T cells, when they get fully activated or hyperactivated, can actually apoptose. I mean, that is one of these sort of immune system regulations, if you will, of how T cells act. So you're exactly right. I mean, I think I would go back to fundamentals of how exactly is the stimulus being applied and what concentration is being used there, and is the whole system sort of hyperactivating the T cells, such as you might be starting to see that apoptosis on the right-hand side, so to speak, of the bell-shaped curve. So that might be applicable to optimize first before testing the agent, whatever that agent is. Yep. So thank you, Wendy. Mm-hmm.
Sure. Moving on to next one. We still have like 5 minutes left to address a few additional questions. One more is question on the T cell polarization assays. Do you look at the expression of unrelated cytokines in the polarized cells, for example, expression of the Th2 cytokines, such as IL-4 in the Th1 polarized cells? Also, have you sorted out the polarized cells and looked at plasticity going forward?
Yeah, this is a great point, but the bad thing is that we haven't done that so far. The only thing for T helper polarization as we do is by the intracellular staining of the signature cytokine. We haven't done the the opposite cytokine profile so far, but this is a good point that since the in vitro assay, both the Th17 and Th2, you can see the L4 and L17A level is really relatively low. So there's still possibility that the cell, majority of the cell will not fully or successfully differentiate into the T helper 2 or T helper 17 cell type. So it is deserved that you do a profiling of other types of cytokines. And in terms of functional study, yeah, we only have done this at Treg function study by suppressive assays, but not Th2, Th1, Th17 so far. But this is a great point, and I think we will further extend our capability and by continue the story and perform more functional study. Thank you for the question.
Thanks. Next one. Does the OT1 EL4 co-culture assay reflect the in vivo efficacy?
This is again a very frequently asked questions. I have to say it cannot fully reflect the in vivo efficacy because OT1 system is especially in vitro co-culture is really simple system in which actually you only have first you artificially stimulate T cell used over peptide at a relatively speaking higher concentration. So the T cell obviously is overactivated, and then you directly co-culture with target cell to provoke the killing. But in vivo things are totally different because the dendritic cell need to migrate, first encounter the antigen and then migrate to the lymph node and then encounter with the T cell, and the T cell responds and then migrate again to the tumor site and perform the killing. And within the whole process, there's a suppressive immune cytokines, suppressive immune cells involved in this process. So the in vivo actually is much more complicated than the in vitro system. So I have to say it is relevant, but cannot fully reflect, the condition of the in vivo system. Thank you for the question.
Sure. So I think we have time for one last question from the audience, and that would be, what is the best assay now to predict CRS or cytokine storm from a potential drug? I think that's a very broad question. Maybe both of you can take some comments.
Thank you for the questions. For in vitro part, actually we have done the sort of cytokine release assays use a human PBMC, and by just directly incubate the PBMC with the tested compound, and to serve as a, relatively speaking, a simple tox or safety study to see whether the compound can exert any immune cell response abnormally by exerting increase the cytokine secretion. This is a pure PBMC based, which is very simple, easy, and fast to do. But again, this is in vitro PBMC assays. For a real CRS prediction, the best way for sure is shift to in vivo model, either in mouse or in non-human primates. Thank you for the questions.
Yeah, Tina, I think you'd answered it. I'm not sure if I have that much more to add. I'm not sure there's a completely predictive or translatable assay from an in vitro setting. The PBMCs, either human or let's say mouse splenocytes in vitro or something, can provide maybe an indication of how much you're amping up or ramping up the immune system to secrete cytokines. But it is only an in vitro system, and it's very different in the actual in vivo setting. So I don't think there is that substitute for doing a tolerability, assessment in the animal once the test article sort of matures to that point in the drug discovery pipeline. Yeah, thank you so much, Andy. Yeah. Mm-hmm.
Yeah. Thanks, Tina, Christine, and thanks, Diego, for joining us for today's webinar. And now we are running out of time. And then thank you, everyone, for attending today's webinar. Now the webinar has concluded. Thanks again.